So, hello everyone. Welcome to the second webinar of the Bheja Fry series. We are really happy to have Dr. Shruti Muralidhar with us.
Yeah, sorry about that. Thank you. I'm really happy to be here. I hope to talk about how the brain is organized, what the brain is, and I am absolutely bowled over by the brain, and I hope to sort of infect you with a little bit of that enthusiasm today. Yeah, like Poonam said, I am one of the speakers who actually appreciates if you put up your hand and ask questions in between. Feel free to use that button there and ask me questions whenever you feel like you want me to stop. So there we go. I see some people testing it, which is great. Thank you so much. Please don't do it again and again because I will stop speaking at that time. Neat. Okay, so today we're going to talk about brains and neurons. Who am I to talk about brains and neurons? I thought I'd give you a little bit of a background on who I am. This is sort of, you could say, my life journey a little bit in academia and in sort of neuroscience. I did my master's in NBRC. I did my PhD at EPFL in Lausanne, in Switzerland. I did my first postdoc at the University of Utah in the US. I did my second postdoc at MIT. And then I'm finally now a research scientist at a small company, a small startup in Toronto, in Canada called Excellent. So the question we all want to ask and the question we all want to answer and sort of we want to discuss is, what is a brain? And I always start sort of talking about the brain, and I always try to start answering this question by asking, who is asking this question? Who is asking, what is a brain? Are you asking a behaviorist? Are you asking somebody who studies behavior? Are you asking them, what is a brain? Are you asking a biologist? Or are you asking a computational neuroscientist? So depending on who you're asking, you're going to get very different answers. And over the course of time, over history, many different kinds of these people have asked this question. And so I'll give you like a small smattering of people who have asked this question. Somebody called William James, he's known as the father of American philosophy. He was sort of a behaviorist and a philosophist, if you could say that, a philosopher. He sort of tried to answer this question as to what is a brain. And he wrote this book called Principles of Psychology, which is probably, one of the basic texts for people who are studying the brain and psychology. So if you haven't heard of him, I would highly recommend that you should go look him up and read about his life and his work. And he had some pretty interesting answers about what the brain is. One of my favorites, because I am a biologist by training, one of my favorite people in neuroscience is Santiago Ramon y Cajal. He is known as a pioneer of modern neuroscience, and I guess you could say he's the closest you can get to a biologist asking the question of what is the brain. He wrote a number of very beautiful books, but also sort of developed theories of how the brain would work. Was one of the few first people to actually get brain tissue in hand and try to study what was in that brain tissue using microscopes, using dyes, using drawings. So he really is sort of a pioneer of modern neuroscience. And if you thought all scientists are kind of boring, this is just like a side note to let you know that he also was a very enthusiastic bodybuilder. So this is one of his photos from way back when, where he wasn't just a scientist. He actually had a full life and developed his hobbies and stuff like that. Computational neuroscience is a complicated it's complicated to ask a computational neuroscientist what a brain is, because they'll have lots of different answers. But here's like a section of a few computational neuroscientists who all contributed in very important ways to how we think about the brain. Louis and Marcel Lapique thought about how a neuron works and how a brain works, and developed things called integrate and fire models. Again, I'm giving you all these names and giving you all these terms not to sort of confuse you, and there is a reason why I'm not going into super detail, because I would like people to sort of take these names and go read by themselves and sort of do your own research, so to speak. Donald Hebb is a very famous scientist who sort of came up with the idea of Hebbian learning, how sort of the brain learns and why we learn. Hodgkin and Huxley are some of my favorite scientists. They are electrophysiologists, and they made a model of an action potential, which is sort of the currency of information in the brain. And Wilfred Roll was one of the few people who started talking about cable theory. So you have lots of cells in the brain, and they all connect to each other through things that look like cables. And so how does this affect the way information is processed in the brain, and so on. So yeah, this is just a cross section. Feel free to use these as jumping boards to sort of read about different aspects of neuroscience, different aspects of neuroscientists and their lives, and so on. So what is the most basic function of a brain, right? I will walk through this like very, very slowly, but it's a really, really cool sort of flow chart. And I think it sort of spans across whether you're a biologist, you're a behaviorist, you're a computational neuroscientist, you're a molecular neuroscientist even. What does the brain do? The brain actually sort of is part of this loop of information that goes in and out of your body, in and out of the environment, your body, and the nervous system. So you have, let's say, an input, any input. It could be, in this case, it's a sensory input. So you see something, you hear something, you feel something. This goes through the body. It's mediated by sensory systems, goes into the nervous system, is processed in some fashion. It's a signal for people who do signal processing, for people who are electrical engineers. It actually does get processed across different levels by different cells, by different groups of cells. Something happens to it, and we call it sort of cognitive processing, and it generates, in a lot of cases, some sort of change in the body that leads to either movement, a reflex, and action that comes as a result of this sort of input of sensory information. This could happen, as you can see, as a reflex. This could happen through voluntary movement, and this sort of generates behavior. It generates sort of responses as an answer to whatever happened in your environment. So in very simple terms, the brain is basically what does all of this processing across species, I would like to say, in most cases. Now, how did they evolve? It's a very complicated organ. It took a lot of steps to get where it is today, depending on which species you're looking at. So it's a very good question to ask, why and how did brains evolve? So over evolutionary time, simple ganglia, and ganglia just are simply like groups of neurons that are just bound together. You get that those have evolved to give you like sulci and gyri in our human brain, which are basically like peaks and valleys of tissue, which are densely packed with neurons. It took time, and along with taking evolutionary time, it took different environments. So depending on your environment, how much computing power do you need? Like, do you simply need to react to something in your environment, or do you need to take that information and process it in multiple different ways and respond in a very complicated fashion? So if you have a very simple environment and a very simple goal, you don't need like a very complicated brain in general. But yeah, if you need to have a wide repertoire of reactions, then yeah, you need a lot more than just a simple set of ganglia. Where is it located? I mean, and why is this important? Because it really is important to know where this brain is located in the body, because it is literally close to the end of your body that leads. So any part of your body that first comes into contact with the environment. So for worms, it's the head. For insects, it's sort of the front bit. For us, it's our head, which has all of these sensory inputs in it, the eyes, the ears, the nose, and things like that. So it's close to the end that leads. It's close to the end that interacts with the environment first. And these are just some illustrations to show you that, as to how you have different animals. So this is basically just like a worm, a flatworm. This is a fly, and this is an octopus. And like similar things, similar sort of neurons have organized in different fashions depending on what this animal is and what it encounters in everyday life. Like a simple flatworm probably doesn't need more than, say, you have chemical sort of cues coming into its body. It has a group of globuli cells. I'll just get to you in a bit. and it needs to sort of generate a sort of response. A fly has a slightly more complicated environment. It flies around. It's very, lots of things coming at it very fast. You need sort of more control. The sort of brain and nervous system are even more patterned and sort of slightly more organized to figure out what to do in these conditions. And of course, octopi, some of my favorite animals, have really sort of, differently organized, complicatedly organized systems. Yes, whoever asked that question. Yes, Poonam.
Yeah, actually, there are a couple of questions in chat, but I guess you presentation mode can't see the chat, so I'll just ask them.
I really can't. No. Can I finish this slide and I'll come to those questions?
I think the questions are from the slide.
Oh.
Yeah. So one person is asking what is ganglia.
A ganglia, like I said, is a group of neurons. It's just a group of neurons bound together. That's a ganglia.
And Karthik is asking any example of organisms with simple ganglia.
Yeah, worms. Worms are a perfect example of organisms with simple ganglia. Neat.
Yeah.
I'm going to. Okay, I'm going to go ahead because I'm sorry if I didn't catch your questions, but I didn't hear anything, so I just kept going. But thank you, Poonam. That was helpful. Neat. Yeah, so location within the body space close to the end that leads. This is like a really great illustration of that. Across different species, across different clades, across different genera, you'd see a primitive plan that plays over and over again, which is that most of the neural tissue, most of the neurons, most of the things that make up the primitive brain are always in the end that leads. And here you can see sort of highlighted in red in different animals. This is where you find neural tissue. This is where you find ganglionic tissue in different species. Sweet. So given that the brain is so varied, and given that there are so many examples of different kinds of brains, then it only makes sense that we should be studying it across many, many different model organisms. And I'll just run through some of these famous model organisms that we use in neuroscience. Like I said before, some of the simplest, some of the easiest to study are worms. This is Caenorhabditis elegans. It is a very elegant model system. We know every single neuron that's in this worm. We know exactly how they develop. We know exactly how they make networks. So it's very useful to study sort of interactions of genes, interactions of network, what will happen if you remove one neuron, and things like that. Flies are some of my favorites because that's how I started learning about neuroscience back in the day. Very cool, again, very sort of simple and easy to understand nervous system, but generates a big repertoire of very complicated behavior. Also very easy to maintain in the lab, very easy to grow, and things like that. So probably one of the favorite model organisms in neuroscience. This is I put this in because it's a classic sort of example. One of the first experiments in learning and memory were done in sort of this animal. It's called the apple snail. Yeah, very, very cool. Again, very simple, very easy. The brain is a bunch of ganglia that are distributed across its body. It's very easy to study. And one of the things that was studied in the context of learning and memory were actually reflexes. This has always surprised me. Danio is zebrafish. It's actually an Indian indigenous model organism. It's found in the foothills of the Himalayas. And now basically the whole world uses it for not just neuroscience, but many, many different kinds of studies too. So I would encourage you to read a little more about the zebrafish and what it is. Mice. This is what I have worked on for a good part of my career. It's sort of really, really, again, not as simple, not a simple brain, not a simple system as the others that came before it, but still a quite complicated system. Very easy to study behavior. So mammalian behavior, and if you want to know how the brain generates mammalian behavior, rats, mice, rodents in general are like a great model system to do that. Yes.
Yeah, Shruti. So some more questions have compiled in the chat window.
Sure. Do you want me to go through them?
Sorry? Yeah, I'll go.
Do you want me to go through them? I can read through them and answer. That's totally fine.
It's all right. I'll read them for you.
Okay.
So Arushi asked, what experiments did you do with flies and which ones? Okay, I believe.
Drosophila melanogaster, and I did a whole bunch of feeding behavior experiments.
And Anushka asked, among all these model organisms, which one do you think are best to study neurodegenerative diseases and why?
They are all excellent. Different diseases manifest differently. They have different backgrounds. They have different rules of why they come about. Depending on what question you're asking, you need to sort of have a good model organism. I would never put one down over the other. So you really need to think about not which model organism you want to study, but really which disease you want to study, and what's the best sort of way to study that aspect of that disease. So yeah, sorry, there's no easy answer on that one.
Okay, one more question by Yashas. How does one determine how much computational power an organism needs? We see a range of computational power in organisms cohabiting in the same environment.
Right, you don't determine it. It's evolution that's determined how much computational power an organism needs. So depending on how comfortable that animal is in its niche, whether it needs to evolve more, whether it needs to get more resources, whether it needs to find a safer place to stay, that's what determines how the brain changes and how it evolves, I guess, over time. Should I go ahead?
Yeah.
I see one or two more.
Yes. So is brain surgery possible in flies?
For sure. That's what I did for like a whole summer. There's lots of labs in India that work on neuroscience and flies. I would encourage you to look them up and try to find out, what they do and how cool it is.
All right. Yeah, I think we can move on now. Thanks.
Neat. Okay.
Well, let me tell you,
Yeah,
madam,
sure.
brain surgery. One of my, I would say, niece's husband happens to be Dr. Chabra. He was at the KGMC Lucknow. He was a brain surgeon, and throughout his life, he must have done a large number of brain surgery. Thanks. This is for the information of the participants. Thank you.
Sure. Okay. Yeah, rodents are some of the model organisms I still work with. So yeah, I have a lot of sort of, love for them, you could say, or sort of I appreciate them a lot more because I actually know how difficult it is and how easy it is to work with them. Cats are like classic. Cats visual system has been worked on by many people in the past. It's harder to do experiments on them nowadays, but we actually learned a lot about how the brain works looking at the visual system of cats for a very long time. Monkeys are some of the favorites, right? So they're the closest in terms of evolution, in terms of brain organization to us. We can do multiple different kinds of experiments with them. You get so much knowledge from just a single sort of monkey, a single sample, that it's amazing. It helps you, having them as model organisms helps you sort of answer more complicated questions and sort of diseases that are closer to humans and human behavior. And finally, humans are not accepted. You can also use humans as model organisms to study neuroscience. There are many different ways to study the brain non-invasively nowadays, MRI, PET scans, and so on. So yeah, depending on what question you're asking and depending on what you want to study, you're going to have to look through the model organisms and see which one actually suits your needs the best. But undeniably, across all these species, we've gotten a lot of information about how brains evolve, about how brains work, about how brains interact with the environment. And so I would never downplay any single one of these model organisms. I really encourage you to go read about all of them because they all have some amazing research work associated with them. Neat. Okay, how is the nervous system organized? Neuroanatomy is hard. I found it very hard when I was in masters. It still, I'm sure, makes kittens and med students cry. It's just hard because it's a complicated organ. It has a lot of working parts, and it's really, really hard. And we made the organization, and we as in humans. And everybody came up with their own names, everybody came up with their own sort of system, and eventually we had to push all of these systems together. And so we have like a really complicated system right now. But what I will do is just try to give you a big view of how the nervous system is organized. And hopefully you can sort of take that as a jumping point and go read on what you feel is interesting. In general, central nervous system, which is basically just the brain and the spinal cord. That's how we look at it. The peripheral nervous system is everything that's not the central nervous system. So everything in your periphery, which is everything outside the brain and the spinal cord. This includes subsystems like autonomic nervous systems, which has the sympathetic and the parasympathetic nervous systems. And now increasingly we're recognizing is that there is an enteric nervous system, which basically is centered around your gut. And that's almost considered like, you know, almost an independent system that works all by itself, but has like a sort of a fast pipe, if you could say, direct up to the brain. So they talk to each other very often and for all important things. How are they organized? This is how it looks like. So you have the brain and the spinal cord, brain up here, spinal cord down your back. This is the central nervous system. All the ganglia, all the nerves, everything outside in your body is the peripheral nervous system. These are sort of like the subdivisions that I talked about. So for example, this is the parasympathetic innervation. This is the sympathetic innervation. I do not expect you to read through this right now and take notes. This is just an illustration to show you that there is a lot of complications involved in how you look at the nervous system, and that it literally innervates every single organ that you have. So the brain does control almost all the organs in your body in different ways with different sort of, you know, with different neurotransmitters and so on. One thing I really like looking at, especially when you're looking at when I'm thinking about neuroanatomy, is the Allen Brain Atlas. And here is sort of like a quick example of what looking at the Allen Brain Atlas looks like. Again, I would highly encourage everybody to go to this website. Just go to Google and type Allen Brain Atlas, A-L-L-E-N, Brain Atlas, and you'll see, you'll be able to see sort of different views of the brain, different stainings of the brain, sections of the brain, things like this, things in 3D. You want to look at just hippocampus, you want to look at just sort of certain parts of the brain, you can. It's color coded, you can zoom in, zoom out. I've been in neuroscience for the last 10 years or so, and I still find it fascinating to spend time on the Allen Brain Atlas website, simply because it's just cool. And it's not just, this is what you're seeing here is basically part of a rodent brain. Here, in this case, is a mouse brain. There's also information for human brains and so on. I'm going to pause here to see if anybody has questions. Doesn't seem like.
So there is one question, is autonomic nervous system the division of peripheral nervous system?
Yes, I guess in a way, yeah, I would say that. It's sort of like a combination between CNS and CNS, but mostly CNS because like a lot of it is outside the brain and spinal cord. Neat. Okay. I am going to go ahead. Sweet. So I'll talk a little bit about what a neuron is, which is basically the fundamental unit of a brain. You're building a Lego sort of castle, you need Lego bits. Neurons are those Lego bits. Most of the pictures you'll see in textbooks look like this. It's one of not my favorite way of depicting a neuron because they look like very many different things. So don't be fooled by what you see in the textbook. The neurons look very different depending on which part of the brain you're looking at. This is one of my favorite examples. This is up in the cortex of your brain. These are layer 5 pyramidal cells. They're beautiful cells that have these long dendrites and sort of this short mat in the bottom. This is one of the parts of the brain I worked on recently, which is the hippocampus. Again, very beautiful sets of cells. In green are just sort of a smattering of cells that are labeled with dyes. This down here, this panel is an example of cerebellar cells, something that not a lot of people study, I feel. but again, some of the most beautiful cells in the brain. And this is what I study nowadays, which is literally brains in a dish. And they look as beautiful, if not more beautiful, growing on a flat dish. And they're all neurons and they all look very different. So again, I encourage you to go and read about different cells, how they look, how do you stain them, and what they're involved in. I think there's a lot of information there if you're interested in neuroscience. Neat. So building blocks of the brain. Like I said, am I okay? Not sure why I heard myself there, but I'm going to go ahead anyway. Building blocks of the brain. So building, like I said, you need Lego blocks before you build a castle with Lego. Two of the most important neurons that make up the brain are excitatory neurons and inhibitory neurons. Excitatory neurons, simple, as they say, as the name says, they actually excite other neurons. So if there is sort of an activation or an excitation in one neuron, its job is to take that activation and pass it on to another excitatory neuron. Inhibitory neurons, exactly the opposite. You excite an inhibitory neuron, it will inhibit activity of all the neurons that it's connected to. Fairly simple. Glia, some of the most least studied parts and cells of the brain. Glia literally means glue. They're what hold the neurons together. They're what supply sort of, glucose and other small molecules to these neurons and keep them running. Many different kinds of glia, microglia, astrocytes, oligodendrocytes, and Schwann cells. This is just sort of a general classification. Okay, so this picture, this is what an excitatory neuron looks like, kind of. These are what inhibitory neurons look like. So the ones in orange, the ones in green, the ones in blue, they look quite different from excitatory neurons, and they're sort of interspersed among each other. Glia, these are what glia look like. You can see a tiny little microglia hiding there. The ones in blue are oligodendrocytes. The ones in green are astrocytes. You can see they contact a lot of neurons and sort of form the buffer between neurons and fill up space in between neurons. And yeah, so we talked about how sort of the brain is organized and what are the components of the brain. How do they all work together? I'm coming back to this sort of illustration I don't like, because it's actually a very simple illustration. It's very easy to use it to explain things. How does information flow within a neuron? And by information, mostly I mean excitation or energy or action potentials and so on. Most neurons receive information via dendrites, which is where the nucleus is. They collect all the information. It comes to the cell body. The cell body sort of does a bit of computation. There's also computation that happens up the dendrites. All of this is then, the cell body, I guess you could say, then decides whether to initiate an action potential or not. And once this action potential is initiated, it travels down this axon to the axon terminal to the next neuron. So in general, this is the flow of information. Like I said, dendrites receive signals, cell body or soma processes signals, axons relay signals to downstream cells. And imagine not one neuron, but multiple neurons in a network. Imagine networks of networks. Imagine huge networks, mega networks together in chunks of brain. You have like a big brain. So it's sort of, you need to sort of start imagining the scale as to where one neuron is and where like a behavior is, which is sort of the ultimate outcome of the brain. I usually talk about famous patients, because if we hadn't sort of looked at people and brains of people in general, we would not have learned a lot about how the brain works. One of the sort of famous patients in neuroscience and psychology, if you want to say, is Phineas Gage. He used to be a railroad worker, and during one of his days, he had a huge accident where a mega long rod went straight through his eyes and outside the back part of his head. Surprisingly, he survived. His brain was fine. He could live a life. He was not the same person he was before the accident, but he actually lived a life. And he was studied by a lot of people, like psychologists, like philosophers, like neuroscientists back in the day. And we got to know a lot about how the brain works. Patient HM, or Henry Molaison, is one of the, again, most famous patients. for looking at learning and memory, sort of diseases, how the brain works around learning, how the hippocampus, which is the part of the brain that's involved in learning and memory, how that works. Because when he was young, he used to have a lot of epilepsy, and one of the things that psychologists and psychiatrists back in the day, neurosurgeons suggested that, oh, we should just remove the part of the brain that's causing the epilepsy. Unfortunately, that part of the brain ended up being like a big chunk of his hippocampus. So it ended up affecting the way he made memory. It ended up affecting the way he made memory in the past, in the future, the kind of things he could remember, and it also affected his learning. So again, famous patients to read about, to sort of delve deep into and understand what we've gained from these people and sort of their accidents, but also like all the amazing things we've learned from their brain. Finally, some of my last slides. I usually like ending this with small facts that people wouldn't have known otherwise. So the brains and neurons are formed from the ectoderm. I don't know if people know what ectoderm is, but it's basically the same origin cells as the ones that produce your skin and hair. So just when you're developing, when you were a tiny little embryo and you were growing up, the same cells that produce skin and hair actually ended up producing neurons in your brain. There's something called neurons are post-mitotic, which means that they won't divide once they're fully mature. There's a lot of sort of back and forth on this, a lot of people working on this, but in general, once a neuron is born, which happens when your brain sort of forms when you're in your developmental stage, they will form, they will mature, and they will stay like that. So unlike skin cells or other cells in your body that die and get replaced again and again, neurons are one of those few cells in your body that don't get replaced when they die, and they will not divide once they're mature. They're one of the few electrically excitable cells in your body, along with sort of muscles to a certain extent and cardiomyocytes in your heart. And they can be some of the largest, longest cells in your body. I mean, there are neurons and axons as long as your legs that go from sort of your lower back all the way down to your pinky toe. So they're really neat. I just think they're really neat, and I hope I've been able to impress on the fact that neuroscience is amazing and neurons are really cool things to study. Students, bachelor students, master students always come up and ask, oh, it sounds like everybody knows everything in neuroscience. No, there are lots and lots of unanswered questions. Developmental neuroscience is a field that still requires a lot more work. We know that neurons develop, we know how they develop, but we don't know what happens if things go awry. So what happens in diseases and so on. We still don't know a lot about neurons and glia and their interactions. So cellular and molecular neuroscience is important to study. Neuroimmunology is an up and coming field, how the nervous system interacts with the immune system. Some of my favorites and sort of things that I was trained in, system neuroscience, is important to study behavior, injury, disease, how the brain reorganizes in response to injury and disease. Learning and memory is a famous field that people have been trying to understand how learning happens, why it happens, and so on. Computational neuroscience, like I said before, a lot of work left to do in modeling how a single neuron works, how a network of neurons works, what happens if you simulate a network of neurons, how do neural networks work, and how do you sort of understand how neurons work by building like a simulation of neurons outside the brain, and so on. Yeah, happy to take any questions, comments, discussions, anything that you have.
Am I audible?
Yes. This is Gagan.
Okay, so,
yeah. Hi.
Yeah, hi. Thank you, Dr. Shruti, for such an insightful session. And so floor is now open for questions. And so should we take from the chat box, right?
Yeah, I can actually go through them. That's not an issue. Let's see. I have Rini asking which type of synapse helps in cognition and learning, electrical or chemical. The answer is both. Both of those synapses are present in your brain and your nervous system. Akanksha is asking, but I guess Vishaka has already answered. Manya asks, could you tell me something about mirror neurons? Mirror neurons are a construct that is actually under research, so it's kind of hard to say something with full certainty, but mirror neurons are supposed to be the neurons that are activated in your brain when you see somebody doing an action that you've done before. in sort of short. I know that's not a great explanation, but it's kind of hard, again, like I said, to explain what it is, because they've been observed in monkeys, but we're not really sure if they're true or not, and there's a lot of research going on in there. Akash asks, people with ADHD, why does ADHD happen? We don't know. I think you should read more about ADHD. Abir asks, optogenetics, yeah, is one of my favorite techniques to use to study the brain. So yes, I think it's a great set of techniques. Sanjay asks, how important is the glymphatic system? I guess it's important because they, develop together. So yes, during development, having glia in the right places, having sort of lymphatic cells in the right places is important, because that's, if they're not in the right place, it would lead to disease, developmental diseases. Rishi, I have no idea what that means. Addiction models for withdrawal study. There's lots of models. Different aspects of addiction can be studied by different models. Saptarshi, what is a neurotransmitter? A molecule that helps two neurons talk to each other. That's a neurotransmitter. Karthik, any research on human behavior based on a particular situation? That's a very broad question. It's a little hard to answer. I would maybe ask you to think about that question a little more and see what you want to know. Akanksha, does new neurons keep developing throughout our lives? So we're not really sure. That is also a point of contention. A lot of studies seem to say that new neurons can be born in the human brain, but there's also studies that say otherwise. So I would encourage you to read about it and see what you can find. It's a developing sort of issue. So yeah. Have I done any intervention? Sorry, no. Abhigna, what are the main limitations we face when we try to transplant even a small part of the brain? Opening the skull and, shoving a piece of brain in there that's not yours. The immune system doesn't like it, among many, many other things. Rimi, have I done any intervention research at UG level? No, I have not. I don't quite know what intervention research means. Shruti, understanding how inputs are perceived. It's both, actually. So understanding how inputs are perceived and processed by the brain, is it being researched at a functional aspect, or is it more of a behavioral studies research question? It's both. Behaviorists study it, neuroscientists study it. Sensory systems still have a lot of unanswered questions. So you need not one approach and not one sort of technique to study it. You need multiple things to study it. Astha, does the glial cell recover if there are any damages? Yes.
Shruti, may I interject? Yeah. So our intended audience is actually the one where we assume no background knowledge, and a lot of these students are who do not know anything. So I guess that's why they are very curious about asking some of the very fundamental things.
Yeah,
so
I'm happy to answer them.
Yeah. So if you could just add in a little bit more detail to your answers, maybe that would help the audience.
That's hard, but I don't know. Okay, let me see if I can add a little more detail.
Questions to cover, but yeah, I think some of them are quite young here, and they don't really have any undergrad science background here.
Yeah. Asta asks, do the glial cells recover if there are any damages in them? Actually, I don't know. That's a good question. Please go read about it. I'm not quite sure. I think there is some amount of recovery as far as I can understand, but in general, I don't think they fully recover. Akanksha, I'm sorry, I don't have time to explain what mirror neurons are, but yeah, if you can find somebody else to talk about this with, I would highly encourage that. Sweet, thanks, Ankush. Nice to see you again. Samar, neurons growing on a flat Petri dish. Yes, it's possible to take neurons out of a brain and grow them on a flat Petri dish to do whatever you want with them. In my case, we test drugs on them and then try to understand how their normal function changes when we throw drugs on them. Arushi, I'm currently not working in India, and I am not working on a project that can include undergrad. Sorry. Sweet. Okay. Do glial cells help in information transmission? It's very highly likely. I cannot give you a textbook level explanation, but basically what sort of current research has shown is that neurons without glia don't develop as fast. They are not as They're not as efficient. They're not as good at, talking to each other. And so having glia just makes everything better. It makes neurotransmitter sort of use better. It makes your neuronal synapses develop faster, the neurons mature faster, their information transfer becomes better, and so on. Neurons aren't capable of multiplying. Yes, that's what I meant by post-mitotic cells. What is the purpose of dural sinuses when there are already venous system in the central nervous system? That is a question you should ask evolution. We study what is there. We try to understand it, but there are some questions that just simply don't have answers. Vaishnavi, what causes an increase or decrease in neurotransmitters? That's a really good question. We don't know. We don't know if epilepsy is caused by change in neurotransmitters or epilepsy causes change in neurotransmitters. So it's a chicken and egg situation that definitely requires more research. What is a neural network? A neural network is a set of modeled neurons, that is computationally modeled neurons that you put together. A neuronal network is something that's inside your head. It's a set of neurons that are connected to each other. What is the current scope and status of research in computational neuroscience? I think it's fantastic. I love reading about computational neuroscience. There's a lot of things left to understand, especially now when every other thing is leading into AI and machine learning. You really need to know how the brain works to understand how to build an efficient system, an efficient neural network. So yeah, there's a huge amount of scope in computational neuroscience. No problem. I'm sorry if I missed any questions in between, but there are some that I just can't answer. That's just the nature of science. And if you need any more clarifications, I guess you can ask it again, or happy to explain deeper.
Am I audible?
Mm-hmm.
Yeah, okay. I think there are no more questions. And if someone wants to ask any question, please, they can raise their hand. And so if there are any questions, okay, so I see two questions. Okay, so Meghna, I think you can unmute yourself and you can ask the question.
Hello, Dr. Shruti. Great talk.
You can call me Shruti, Meghna, no problem.
And amazing work you do even with BiasWatch India. And I wanted to ask one thing, like you're trained as a neuroscientist in a very academic way, but how different is it to work in a firm? Like I'm kind of thinking whether to shift to not work in a necessarily academic setting. So how did you navigate through that? I know it would be a lengthy answer, but I just don't know many women who have transitioned. So can you just tell a bit of it? And thank you.
Sure. Yeah, no problem. So the transition is not easy. It takes a lot of sort of courage on your part, especially if you've stayed in academic neuroscience for a long time. It takes a lot of courage to think outside the box and try to, use what you've learned and apply it to a different situation. But I don't think, I don't think moving out of academia is bad at all. It's good for who you are if that's what you need at the time. The settings in the sense are, a few things that I can touch on. Academia, money is less, but you have a lot of time. Sort of industry, there's a lot of money, but you don't have a lot of time. So it's a different pressure. It's different goals. So if you can sort of adjust to one or the other, depending on what your strengths are, that's the best thing you can do. There was one more hand up. Saptarshi.
Yes, Saptrishi. Saptrishi, if I'm pronouncing it right, so you can unmute yourself.
Go ahead.
Yeah.
I don't hear you, Saptarshi. Maybe we should go to the
I think
next.
Yeah, I think we can take another one. I see Uh,
I see Yashovardhan.
Yashovardhan. Yeah.
Yeah, hi. So, adding on to Meghna's question, like how different is academic research from the one we do at firm in terms of flexibility and the experience?
Well, clearly, in a company, you have a project and you're working towards a very specifically defined goal. In academia, that happens, but a little less rarely. Well, I guess a little more rarely. If you're on a project that you know has to have a definite endpoint, like a master's thesis or a bachelor's thesis, yeah, you can sort of work towards the goal and be done with it in a year, 2 years, what have you. But things like postdocs and PhDs are sort of more open-ended. So you need to define your goals. You need to, figure out what you want to do. So in that way, academia is a lot more open-ended, and sort of industry is a lot more structured in terms of goals and what you need to achieve.
Okay, so like one more question in addition to that, I mean, roughly figuring out what would be the prerequisites for someone to go into firm research, like in terms of their like educational achievements
I would
or
say
the
you can
degrees.
start applying for jobs right after a master's. See which companies require masters in neuroscience, masters in allied fields. Look for job descriptions that say the same.
Okay, thanks a lot.
Yeah.
Hema Sankharva, maybe you can unmute yourself and ask the question.
Yes. Hello, ma'am. Good evening. I come from chemistry background, and I wanted to ask, like you talked about the computational neuroscience. So, if you could explain or give us a few examples, like how we can know more about it. Like, if someone wants to make a career into it, like what are the courses or what are the sources we can learn from?
You can learn anything, or you can read any book that talks about neural networks. There are, I don't know specifically the courses in India, but I guess any sort of neuroscience PhD level or graduate level course is going to give you training in the basics of computational neuroscience. But yeah, there's lots of books that you can read. I don't have any at the top of my head right now, but yeah, anything that talks about neural networks, anything that talks about simulation, anything that talks about modeling of neurons and neuronal activity is a good start.
Okay, ma'am. Thank you so much.
Okay, so someone replied like graph theory would help you as a starter. So maybe that could be helpful, Hima. And so we have another two questions. And so before
I
your
can take
master's
those.
in and yeah.
Yeah, I did. I was going to put that in, but we kind of had a link glitch in the morning. I did my bachelor's in Acharya Narendra Dev College in Delhi University, and I did it in biomedical sciences. And any connection of glia cells with eyes, yes, there are lots of glias in the retina. I'm not quite sure where that question is going, but yes, there's lots of glia there.
I think if someone wants to ask a question, they can please raise your hand. And so I think there are no more questions in the chat. So I think we should wrap it up. Okay, so we see one hand, Akanksha. So yeah, please go ahead.
Hello, ma'am. Ma'am, you mentioned about enteric nervous system. Will you tell a bit about it?
Yeah, I don't know that much about it, to be very honest, because I have not studied it at all. But as far as I can understand, it's basically a huge network of neurons that sort of encompass the gut, lots of your colon and lots of the stomach, and they're responsible for a lot of things like release of acid, the timing of the release of acid into your stomach, how your colon and how sort of the large and the small intestine move to push food in and out, well, I guess only out, not in, through the colon for better absorption. For a long time, we thought that these neurons weren't important and all they did was just, do this movement and sort of do some other basic functions. But the more we study it, the more we figured out that this enteric nervous system is an actual entity. It's actually a thing by itself, and it directly talks to the brain, and it sort of has neurotransmitters that are common with the CNS, and has a lot of effect on how the brain works, and of course also is affected by how the brain works. So yeah, that's pretty much the short answer.
Arushi, maybe you can go ahead next.
Hi, am I audible?
Yeah, go
I
ahead.
wanted to ask, before your master's at NBRC, where did you do your bachelor's and what was the course?
Just answered that. You asked the question in the
Oh, you
chat.
did?
I was at Delhi University. I did biomedical sciences in Acharya Narendra Dev College.
Okay, cool. Thanks.
Sure.
Okay, I think we have answered all the questions. Okay, so there is one more in the chat box.
Avir, that's great. Get a basic grounding in biology, I would say, if you want to study the brain. I don't think, I'm not a career counselor, so I can't tell you whether you should go for BSc or BTech, but go to a course that speaks to you. Go to a course that you think you can learn the max out of. I think Akanksha, do you have a question again? No. Mariam, go ahead.
Hello, ma'am. Ma'am, I want to ask, we are from bio side students, so we often feel like, do we need to learn the programming and all of such skills? So where do we begin?
I would
Because
say yes.
there's a
Go ahead.
because the programming that I need in bio informatics kind of a lab. So how much of maths you have to learn? How much of computer science you have to include? Can you give like a brief idea?
It really depends on, again, I know I keep saying this and I feel bad saying this, but it really does depend on the question you're asking. If you're working in a lab that works on genomes, yes, you need to know a little bit of coding because you need to do genomic analysis. If you're working in a lab that does, just behavior, yeah, even then you're going to need a little bit of coding. So in general, I think my sort of big answer to you is yes, coding is going to be useful irrespective of what you're doing in biology, for analysis, for organization, for many, many different things. But the level of coding you need is going to depend on the kind of question you're asking. If you're asking something really complicated, you're going to need to understand a lot more of coding to sort of fix that problem. If that problem is not that difficult or it's sort of not that complicated, you might just need to know basic coding. So I'm sorry, that's a very open-ended question and I can't really give you a specific reply. It really depends on the question you're asking.
Thank you, ma'am.
Sure.
I think there are no more questions. Yeah, so thank you, Dr. Shruti, for giving us speaks okay, so yeah, just a minute. Am I audible?
Yes, I see more questions coming. I'm sorry, guys. This was fun, but I also need to do other things. So
Okay.
I'm happy to keep talking. I mean, I don't have time, but please keep talking with yourselves and try to find answers that you need. Yeah, thanks, Gagan. Not a problem.
Yeah, okay. So yeah, thank you so much, for giving us all the peeks into brain evolution and introducing us to different model organisms and neurons and glia and all of this. It was like a much needed session for especially the young people. So I now invite Dr. Poonam Thakur, the co-founder of Mind Gala initiative, to address the gathering and deliver the vote of thanks.
Okay, we are not going that formal with this webinar series, but thank you, Gagan, for taking up all the questions. Thanks a lot, Shruti, for accepting our invitation. It was really nice to have you here. And yeah, the idea to get Shruti on board was, we can get out of the academic labs and still talk about cool neuroscience, and introduction to neuroscience should be had had from all perspectives out there. And I'm really glad that a lot of audience members actually related to that, they had questions about that. So for audience members who would like to continue these discussions or questions further, I would highly encourage you to go and join Project Encephalon. They have a Slack channel. Sorry, not Slack. There is another platform channel. Just go to the website, you'll find it. There you can find a lot of such kind of mentorship available to you. And you can keep asking all of these specific questions from other people who are actively engaged in neuroscience research in academic settings majorly. So thank you everyone for joining in evening, and it was really nice to have you all. And we would be back soon with another webinar to let the brain blow your mind. All right.
Thanks everybody. Have a good evening.
Ma'am.
Bye.